Hi, my name is Fragkouli. I'm a PhD candidate at the University of Athens and also work at the Institute of Applied Biosciences at the Center of Research and Technology in Thessaloniki, Greece. Today, I'm going to present to you our work with title Synthetic Genomics Data Generation and Evaluation for the use case of benchmarking somatic variant calling algorithms. So the challenge that arises with somatic variant calling is the absence of standards and criteria for reliable and consistent identification of variants. Thus, variant calling may lead to largely different results when using different solutions, mainly to the differences in their algorithmic nature, which result in diverse outcomes. Things get more complicated when dealing with variants at low frequencies. The absence of a ground truth becomes obvious when playing around in this field. For this reason, our initial focus was on generating an in silico dataset with known variants, which could act as ground truth and be used for benchmarking variant calling algorithms. To achieve this, the algorithm NEAT was implemented, and based on the gene TP53, we began generating genomics synthetic data. One thing that we couldn't give as input was the specific allele frequency of these variants. So to overcome this, we made 10 independent runs and produced 10 bump files, each of 500 coverage. Then we identified the chromosomal position of all the variants that were at 100% allele frequency in each of those files and defined them as gold standard variants. Moving on, we merged all these 10 files into one final bump, which had now coverage of 5,000, and the allele frequency of our gold standard variants would become 10%. Then it was time to pick our candidate variant caller to test this dataset. For this purpose, we chose the somatic variant calling algorithm GATK-MUTEC2, to which we gave our BAM file of 5,000 coverage as input in order to identify the somatic variants that were present in this file. The overall results can be seen in this slide. As you can see in the left figure, the coverage reported by GATK has a mean of 1,000, while the ground truth was designed to have 5,000. This happens because of the downsampling that GATK performs that results in misrepresentation of information. Of course, this also affects the allele frequency of the detected variants, as depicted in the middle figure that shows the bar plots of the allele frequency. As you can see, there are a lot of differences in the values of the ground truth and those reported by GATK. On the right, you can see the density plots of allele frequency for each of the 4 letters that represent the DNA basis. The top panel corresponds to the ground truth and the down panel to the GATK. Here we see the SNPs and their corresponding allele frequency on the left. The green color represents our ground truth and the red color represents the results that were produced by GATK. We see that the variant caller missed a lot of the variants and in many cases reported a different value for the allele frequency of the detected ones. On the right, there is a Venn plot for all the variants in both ground truth dataset and from the results from GATK. We see that their agreement is about 30%. So the overall highlights of our work is that we generated synthetic genomics data based on a certain gene and defined a ground truth set of known variants that would then offer the chance to perform benchmarking with confidence. Finally, we investigated the impact of variant callers in variants at low frequencies. So if you're interested in learning more about our pipeline, please scan this QR code to go to our GitHub repository. The poster with our results, thoughts, and comments is going to be up on Monday, 24th of July during poster session A with number 102. So if you find all this effort interesting, feel free to come say hi and have a chat with me. And if you're not attending the conference, you can find my details and my email on the bottom left of the slide. So thank you very much for your attention and have a nice day.